So this molecule of DNA, this is what was passed down to you from your parents. Inside the sperm cell and inside the egg cell were molecules of DNA with these chemical letters, these T's and C's and G's and A's, and that's how you inherited your genetic information. It's that DNA code that determines whether you have blue eyes or brown eyes, whether you're tall or short, and as I said before, whether you're a human or an earthworm or an oak tree. And you may wonder, how is that? If every living cell, if every species on Earth has the same four letters in its genetic code, then how can we have differences? If I have the same four letters in my DNA as you do, then why don't we look exactly the same? And the answer is pretty simple. It's the sequence of letters really that matters. So just to make up a simple example, this particular sequence of T, C, A, C, T, T, G, G, T, C, maybe that's the gene for brown eyes, which is what I have. But if we were to change just a couple of letters, maybe this is now G, G, A, C, T, T, G, G, T, C. That might be blue eyes. Or change a couple of other letters and maybe it's green eyes. And I'm just making up this example, but that's the idea. Is the sequence of letters is really what determines the characteristics that we see in the organism. It determines how we look, and it also determines whether you're one species or another. But the one thing that all organisms definitely do share in common is they all use this same molecule and these same four letters to store and transmit genetic information. So finally, we've reached the end of our list of 9 characteristics that all living organisms share in common. So as different as you may think you are from a bacterium, an oak tree, or a mushroom, and you certainly are different from those organisms in many ways, but it's important to remember that all of those organisms, including you, use DNA to store and transmit genetic information, can maintain homeostasis, are made of cells. The bacterium is made of only one, and you're made of trillions, but you're made of cells, one or more cells. All living things can reproduce. They can grow and develop. They can obtain and use energy, respond to stimuli, are highly complex. Remember those multiple levels of complexity that build on each other. And all living things can adapt. Remember the adaptations that we saw with the snowshoe hare and the cactus. So all living things share these 9 characteristics in common. All right, shifting gears again, as I bring this introduction to biology to a close, I thought that it was appropriate to discuss a little bit an instrument that's very important in the study of biology. And you're looking at it here. It's called a compound light microscope. Now, compound means more than one. And the reason we call this a compound microscope is because anytime you're looking at a specimen, like this microscope slide here, some kind of, maybe they're looking at some kind of tissue or plant cells or animal cells or something, whatever's being viewed is being magnified twice. There's a lens up here in the part that you put your eye close to called an ocular lens. It's in the eyepiece. But then there's another lens down here a little bit closer to the specimen that's called an objective lens. And anytime you look through a microscope, the image is being magnified twice, once by this lens and once by the other. So you've got 2 lenses magnifying the object at any given moment. That's why it's called a compound microscope. Now, it's also called a light microscope because in order to see anything, light has to pass through the specimen that you're trying to view. And modern day microscopes have a power cord and some sort of electrical, usually a fluorescent light bulb that you can switch on and off with the power switch here. The old-fashioned microscopes simply had a mirror that you would have to sit next to a window or have a lamp shining down, and you would use the mirror at the bottom of the microscope to shine the light up through the specimen. But one way or another, you have to have light passing through in order to see anything. Now, at some point in biology class, you'll have an opportunity to use the microscope, and it's important to know the parts so that you can learn how to use it properly. So some of these I've already mentioned. The eyepiece is the portion of the microscope that you actually look into, and it has a lens inside of it called the ocular lens. And if you, I don't know how well you can see this, but it tells you here in the fine print that this particular lens has a power of 10x, which means it magnifies, makes the object look 10 times larger than it really is. The other type of lens, remember, is called the objective lens, and most microscopes have 2 or 3 or sometimes 4 different objective lenses. They're different lengths and have different powers of magnification. And you can rotate this piece here called the nose piece to select, in this case, low power or high power on your microscope. Okay, the next part that I want to discuss is called the arm. This whole back portion is called the arm of the microscope. Anytime you're carrying a microscope, you should have one hand on the arm and the other hand up underneath the microscope. This part down here is called the base. You always carry it with 2 hands. These even a relatively inexpensive microscope costs something on the order of $300 to $400. I think the ones that we have here at Union Pines are about $500 a piece, and so you don't want to drop it. So one hand on the arm and the other hand on the base. Most microscopes have 2 different focusing knobs. The larger of the 2 is called the coarse focus knob or coarse adjustment. And when you rotate that knob, depending on the microscope, it will either cause the objective lenses to go up and down, or it looks like on this particular scope, what actually moves is this part here. It's called the stage, which is the little platform where the specimen is located. Rotating that coarse adjustment knob will move that stage up and down, and that allows you to bring the image into focus. The smaller knob is called the fine focus knob or the fine adjustment, and it does just what it sounds. It makes very, very tiny adjustments in the focus. When you are on a high power lens, if you're using, say, this high power objective, the fine adjustment knob is the only one you should ever touch. The coarse adjustment really is just for low power magnification. Once you move up to high power, you only need to make very fine adjustments using that smaller knob. I've already mentioned the base is the bottom of the microscope. Just underneath the stage, underneath this platform, you'll see a little rotating disc. You can barely see it here. That's called the diaphragm. And as you rotate that, as it turns, there are holes in the diaphragm that let either a lot of light pass through or a very small amount of light. And what that allows you to do is to regulate whether the image is very bright or very dim. And you might think, well, golly, I would always want the image to be bright, but sometimes you can actually see things more clearly in dim light. And so the diaphragm allows you to regulate that. The stage is simply the platform where you place the specimen that you're going to view. Okay, so in our final segment, we'll discuss how to calculate the total magnification that you're using at any particular moment with the microscope.